Right, so hi everybody, welcome to this last class of genomics. It's a real pleasure to be here, in a sense, even though we're back online. Very unfortunately, I kind of start to hate my little screen. I've seen it way too much, and I would really like to be in the room with volunteers, those who want to. But that's the way it is. So I've decided to do it online. I have no regret, but still, my wish would have been that it would be in person. So before we start, I'd like to go back on some things that came up in the quizzes in general, because I think it's interesting for the purpose of learning. So, linkage disequilibrium. So I knew this would be hard, and this was hard. And if you still have problems understanding it, then it's normal. It's just because you're normal. There's no way you can understand this in just 1 year. In principle, it takes at least 2 consecutive years or 2 consecutive semesters of learning, let's say, before you can really wrap your head around the concept of linkage disequilibrium. So there was this question in the quiz that really almost everybody got wrong. There was 37 wrong answers, and the question was a simple one, which is linkage disequilibrium means that there is less recombination between the two sites. And of course, that was a question that I knew would create a lot of confusion, and it was there for that reason. Just to make you realize, no, that's not the case. This is not what linkage disequilibrium is. I know that it's the way that many people think about it. I know that even for some teachers, this is what they're going to say, but that's not true. Even teachers need to understand linkage disequilibrium. So why is it not true? It's not true because of the slide that's here, if you remember it, from the lecture where I first introduced linkage disequilibrium. So in that lecture, I introduced the concept first, and we had a break, and right after the break I came with the concept again, and this time I presented it this way with that example. So I'm going to go through that example again to highlight really why it matters. So linkage disequilibrium just means that the genotype at one locus, that is to say a pair of alleles, is correlated with the genotype at another locus, another pair of alleles. And I give the example of this migration where people would come from another region and then would suddenly make a merger with people from that region. And in this example, the alleles are not even on the same chromosome. Okay, they cannot be recombination between them. They are not on the same chromosome. And so at the generation 0, so of course there is linkage disequilibrium, because if you know the genotype at this allele, if you are pale allele, then you also know the genotype at the other allele, you're also pale. You could say, okay, well, that doesn't count because they haven't mated yet. Okay, so let's wait 1 generation. So after generation 1, you can make only 3 combinations. Okay, either you're homozygote, double homozygote the other way, or double heterozygote. You cannot be anything else. All right, so if you combine these genotypes that are on the left, you can make only these 3. So if you know that you're heterozygote at the orange locus, you also know that you must be heterozygote at the blue locus. Okay, so the knowledge of the orange locus gives you complete knowledge of the genotype at the blue locus. So you have perfect linkage disequilibrium. And yet the genes are not even on the same chromosome. So it's got nothing to do with recombination. Okay. Question from Andrew: how does the word disequilibrium have any meaning then? What is not equal, or what makes it in equilibrium? That's an excellent question, Andrew. So the equilibrium is in terms of the frequency of the alleles. Okay, so like the pale orange allele has got frequency 1/2. The pale blue allele has got frequency 1/2. And so you have equilibrium when the frequencies of all the possible genotypes are the products of the frequencies of the individual genotypes at one single locus. Okay, that's equilibrium. That's in the other slide that I've presented about this. So when the frequency or the expected frequencies, okay, then you have equilibrium. But here at that point, you don't yet have the expected frequencies. So that's why you can call it disequilibrium. So the equilibrium has got nothing to do with recombination, with an equilibrium at recombination. It's a statistical equilibrium. It's not a biological equilibrium. Okay, that's what's confusing. And of course, I mean, I was confused for several years with that. I would also have said when I was a student, yeah, it's possible that it means that there is less recombination than expected, but it's not what it means. Right? The loci do not even need to be linked on the same chromosome, so that was in the slide to prime this idea that it's got nothing to do with recombination, it's got to do with statistical frequencies. I'm not going to insist, but I know that this has been extremely confusing for some people, and they maybe even thought that it was a mistake in the quiz. No, it was not. Like, it really is not the fact that there is less recombination than expected. If you still have problem with the linkage disequilibrium, it's normal. Like I said, it will take at least 2 semesters before you can really understand that properly and make fewer mistakes with it. Another one that I wanted just for the fun to get back to, so there was only one person, of course, who gave the right answer to that question. It's the most wonderful trap ever made for the human mind, the question about the siblings of Albert. So you meet a woman who has 2 children. So at that stage, what you know is that there are 4 possible cases, right? So either she had a girl and then a girl and then a boy and then a girl, or a boy and then a boy. And if you look at this, like, this is perfectly symmetric. Everybody will agree with that. So you have 4 cases, equally likely, even though the probability of a boy being born is slightly higher than the ones for girls. Let's consider that this is all equally likely. So she has a son, Albert. So case 1 is not possible. When she tells you that she has a son who's named Albert, she's just telling you that case number 1 does not exist. So you have case number 2, 3, and 4 only. The only case where there are 2 boys is number 4. So that's 1 case out of 3. So the proper answer to that question, even though I know it's the most surprising thing ever, is 1/3. Okay, so when she tells you that she has a son who's named Albert, suddenly, as soon as she says that, the probability that Albert has a brother is 1/3. It's not 1/2. And you have to be really careful with the way things are phrased when you tackle these kind of problems. I know this has nothing to do with genomics, but it's fun anyway. So if she says her oldest child is a son, so it removes case 1, girl, girl, but also case 2, girl, boy, because the oldest child here is a girl. So now you have 2 cases, 3 and 4, and in only one of them you got boy, boy. So now when she says that, the probability that Albert has a brother is 1/2. And if she says that the youngest child is his son, it's the same. It's 1/2. So details matters exceedingly for these kind of questions. And I know that some people don't believe that the probability is 1/3. They say, no, that's a trick of the mind. If that happened in reality, it'd be 1/2. No, that's wrong. It's really 1/3. No kidding. Shouldn't the answer be 1/3 or 1/2 because of the ambiguity? No, she really says that she has one son, Albert. She doesn't say that her oldest son is, her oldest child is a son, or her youngest child is a son. She says that one. So the answer is really 1/3. It really depends on what she says, basically, because she could say that in both cases, but she could not say that all the time, right? So, like, in these cases, the details really matter for these things. I know it's hard. I wanted to use that example to show you that when you work with probabilities, we don't have good intuitions. Right? Like, if you use intuition, you're constantly wrong about these things. You really need to write it down to go through all the cases. Intuition is your worst enemy in that case. Blog post stuff. So very soon, not very soon, it's at the very end of the semester, as soon as we're done with the exam. So I have moved on Quarkus the deadline for the final exam to stick with what the registrar said. So after that, then will be the feedback sessions. So the day after the deadline, Michelle will send you an email with a link to a shared Google Doc where you will post, where your post will appear with 3 other random posts. Okay, we'll make groups of 4 posts. There will be yours and then 3 others that were assigned at random. You can comment the Google Doc. Okay, you will have privileges to comment the document, but you can't edit. And you will have to give feedback on the other post with a template below. So this is already on Quarkus. The assignment is already on Quarkus. So you have to follow that template. If you do not follow that template, you will not get the points for the feedback session. So you have to comment, there's your post, you have 3 other posts to comment on, so that's 3 bonus points, 3 points. You have to have a 3 to 5 sentence summary of the post. That's to make sure that you're giving feedback about what you understand to the person who wrote. If you understood nothing, the person says maybe it's because it didn't explain that very well. A minimum of 3 positive comments. We need to be very kind and very nice to each other, and there's always positive things in everything. 2 concrete suggestions for improvements. We all do things that need to be better. And looking for typos, you have to find a typo. If you cannot find a single typo, then you have to say, I did not find a typo, but you have to try and find one. Is there a maximum of how many posts you can comment on? In practice, yeah. In practice, it's gonna be 3 because you won't really have access to the other posts. But what I'm gonna do at the end is I'm going to open a Google Doc which you can edit. And there you can put your post if you want. So that everybody can read your post if you want to share it with the others. So you have to share your post with at least 3 other people. If you want to share with everybody, then I'll just make a Google Doc that you can paste your post to say that's what I wrote, and then you can read other people's posts, and there you can make the comments you want, but you won't get bonus points for that. Okay, so you get +1 point per post you comment if you follow this template. If a group is smaller than 4, I will look for volunteers to fill in the groups, but it's volunteers, so you also not get points for being a volunteer. So it's 3 points, not 1. If you submit your final blog post late, you will not be able to participate to the feedback sessions. Okay, I explained that I have to work really fast after I got the final blog post because at the end of the semester I have to close everything, etc. I won't have time to wait too much for latecomers. If you're late, then you're late. As usual, the jokers will apply, but you will miss the feedback session. Question from Uyen. Since we have feedback form from our peers after the submission deadline, do we make adjustment accordingly, or is it just for future improvements? It's just for future improvement. That's an excellent question. It's also for those of you who want to put your post online. Okay, like that, okay, if I want to put it online and 3 people tell the same thing, like this is not very clear, that's a good reason to maybe fix it. But it's for 2 reasons, just for you to improve and to just see what you could do better next time, but also for you to see what the other have done. Right? That there are 3 posts that you can read. That's also quite interesting, just for the sake of knowing what they have done. So there was a release of a new post just on Monday. It's the first Monday of every month so far because we had 12 posts. And it's a very interesting post. It's one of my favorite, the one that was published. Consider subscribing. You can see like here, there is the front page of the Genomic Odyssey. That's the blog post. There's the subscribe button here. So the blog receives more attention if it has more subscribers and if the posts are liked. Some of you have subscribed. I think it's a good idea. Thank you for doing that. Also, the more you share the posts, the more your own post will be read. So you have to understand a little bit how these things work online. So many people find posts because they Google it. And so you want the Genomic Odyssey with your post to be kind of high in the Google ranking. And that is not just based on the content, very far from that. There's an algorithm that's called PageRank that's counting the number of links that point to the Genomic Odyssey. And the more links are out there that point to the Genomic Odyssey, and the more it's going to increase the rank of everything that's on the Genomic Odyssey. So if you increase the number of ranks for other people's posts, you are increasing the rank for your own post. Like it's just like everything on the Genomic Odyssey will be considered the same by Google and all the other search engines. Okay, and on top of it, the more likes, this is using WordPress, and the more WordPress equivalent of likes are there, and then the more the blogs go in the ranking. So this is up to you, of course, not forcing anyone to do that. But if you plan on submitting your post online, it's a good idea. It's a good investment to just subscribe. That'll boost a little bit the post on PageRank, and also to link to the post you like every now and then, just to make sure that there's a constant influx of links that point to the Genomic Odyssey. That's the way things work in the online world. I don't want to make you too worried about this. This is not super important. What matters is, of course, the quality of what you write. But in case you want to be read, you have to be aware of how these things work. And there's a bit of a karma. You know, the more you share, the more you receive. So that's it for the peripheral type of information. So now we're going to jump into one topic, which is quite interesting, that has come up as one of the most important topics in the last 10 to 15 years, and that's epigenomics. So what is epigenomics, and what's the main question of epigenomics? One of the things that really is the most striking when you look at a body like the human body is how to maintain cell identity. So these are examples of cells from the human body, like the motor neuron, red blood cell, bone cell, cells in the inner lining of the intestine, or the white blood cell. And you can see, of course, they give them different color. I'm not sure that they have these colors, but their shape are really completely different, and their functions also, of course, completely different. So the cells of the body are very different, and yet they have exactly the same genome, almost exactly in the case of the B and T cells. So how is it possible to maintain such a diversity from the same genes? Why don't cells spontaneously change identity? I hope you see the problem, right? Like, they all have exactly the same DNA, except the B and T cells, and that's just a tiny little difference. So they have exactly the same information to start with. Through development, they will take different shapes, different functions. But how can they keep it, right? So what is it that says that this red blood cell is always going to be a red blood cell? Why does it not suddenly start to become a bone cell or something like that? There is a memory. There has to be a memory that tells this red blood cell, you know, you're a red blood cell and you're not going to be something else. And why is it possible that when a red blood cell divides, it never, ever, ever gives a neuron? That just never happens. So how is that possible? How is this information transmitted from the parents to say, we are in the red blood cell lineage and we're never going to make neurons, ever? How is that even possible? There's also the question of developmental paths. So the environment plays a key role in shaping organisms. Depending on what happens early on, development will go a certain way or another, and yet the organism has exactly the same genome. Ants give a spectacular example of morphological changes that are mostly based on the kind of food that the embryos eat. So you can see that these ants here have approximately the same genome. So ants, just like bees, share more than 50% similarity between them. They're like 75% similarity. So it's almost exactly the same genome, and you could even make clones. It doesn't matter, like, it'd be the same. Like, with exactly the same genome, if they eat royal jelly, then they'll grow a queen, and if they're treated one other way, they'll be a soldier, otherwise they'll be a miner, and that's a male here. So of course males have not exactly the same genome, that's fine, but these different shapes and forms here have got exactly the same genome. So you got so much diversity just based on an event that happened early in the development. So how do you keep memory of that event in the adult life? How is that possible? So it cannot be genetic. All right, that's a print, that's an imprint on from the environment on the organism, and they're going to keep that imprint for the whole life. And that imprint is not in the DNA, it's somewhere else. So where is it and what is it? I hope that the question makes sense for you. So many people have worked on that question, starting from roughly the beginning of genetics, actually. So as soon as people started to understand how genes work and what they're made of, then they understood that they don't explain everything. And there's this very famous picture that maybe you've seen multiple times by Conrad Waddington from a paper from 1948, if I remember, where he made that picture of a landscape. And so that's the way he views the way things work in the organism. So he thinks of development as a ball rolling down a slope. And you can see the slope has got some valleys and peaks. And so it's random whether the ball is going to go left or right here. And after this is going to be again random whether it goes left and right, and so on and so on. So each ball is a cell, if you want, and it's rolling down a slope. So it can go only one way, it cannot go up, okay? And there will be random bifurcations that are going to allow it to turn into one type of cell or another type of cell. And so that here is the development, and there's a random component. If you have enough balls, of course, you're going to explore all the possible paths for the development, and that's why the organism is balanced, according to him. But what makes the hills and the valleys is the genes. So he made this other picture that is a bit less famous, but that's still figure 5 from the same paper, where he says, now let's look under this landscape that's here. What would we see? We would see kind of bolts like that, and these are the genes and the bolts of their interaction. So what determines the landscape is genetic, but once this landscape is determined, it's only a potential. So then it depends, it will be randomly whether cell will be that or that. Of course, if you have a mutation that makes a hill disappear and turns a hill into a valley, then the outcomes that were possible with one genotype are no longer possible, or vice versa. And so the way he sees development with this picture is that you have to think of genetics being just a proposal, and development being the either accepting or rejecting the proposal that is made by the genetics. So there's no mechanism here. Okay, you understand that this is like very hand wavy drawing with with analogies, etc. But there's still an analogy that's very, very much in use today. This one of the Waddington landscape, and has kind of inspired the imagination of biologists to that day. That's why I wanted to show that to you. But there's no mechanism. People say, yeah, it's probably the way it works. It's a good analogy to think about development and to think about these epigenetic phenomena. But what are the mechanisms? How would that work? So one example of something that's almost completely understood is the X chromosome of female mammals. So it's almost completely understood. There are still many, many questions, but that's the archetype of an epigenetic mechanism. So I'm going to tell you a little bit the story of what we know about the X chromosome inactivation in females. I'm going to go relatively on the surface only, not going to go too deep in the mechanisms. But what I want you to understand from that example is that that's really the archetype of how things work for epigenetic mechanism. So the first thing that was kind of a surprise was the discovery of the Barr body. That's something that maybe you've seen in the textbook or in the other classes. So Murray Llewellyn Barr, who was actually someone working in Ontario, I learned that the other day, discovered the Barr body. And so he stained the DNA of cat neurons. In 1949, he accidentally discovered a so-called nuclear satellite. So you see, he was just looking at neurons of cats for completely different reasons. It has nothing to do with even formal genetics. He was working for the army to understand how pilots had pressure when they were in the planes. And so they thought, maybe you just look at their neurons, but we can't look at their neurons, so we're going to look at the neurons of cats. And so in some cats, they saw this tiny little dot here. And so what you see here is just DNA, and what appears in black are the places that don't have a lot of DNA. So that's the nucleolus here. It doesn't have a lot of DNA inside the nucleus because it's got a lot of RNA. Okay, so when you stain DNA, in general, the nucleolus appears like a little empty space. And there was also this little dot that they could see sometimes, but not always. And so in the beginning, they thought, okay, maybe it's when we stimulate the cats, when we just put electric shocks in them, or when they're afraid. And so this little thing here must be some response that they had to whatever treatment we imposed on them. Turned out it was nothing like that. It was just females. At some point they realized, okay, every time we watch a female neuron, then there's this little dot that's here, and when that's a male, this little dot is gone. So they called it the nucleolar satellite because it was always really next to the nucleolus inside the neurons. What you see here is the nucleus. And now it's called the Barr body from the person who noticed it first. Okay, this is now referred to as the Barr body, which is one of the two X chromosomes. In the coming years, they figured out what is this, and then within 10 years, they had already understood that that little thing that you see here is one of the two chromosomes that are present in the female cells of the cats. So the next relatively big thing that happened is that Mary Lyon, that person here, when she was more or less as she appeared in 1961, she died recently, hypothesized that one of the two X chromosomes of females was inactivated at random in the early embryo, meaning that females are chimeras in the genetic sense. So Mary Lyon was studying the color of mammals, and she was really interested for very general genetic purposes in genes that would be on the X and that would affect the color of the skin or the color of the fur, if you want. So she was studying these mice, and she was really trying to find these mutations or these natural variants that would have different colors depending on the sex. You know, that's female and males would have different colors. And then she found some stuff. But basically, it was purely based on, you know, a hypothesis. To be honest, the evidence that she had to propose that was actually very, very small. She just connected some ideas that were in the air, and she said, look, one way to explain these phenomenon is that perhaps one of the two chromosomes just goes randomly extinct. Okay, and so then if that happens early in the embryo, that will create a patch of cell that has one chromosome active that would give a certain fur color, and another patch of cells that has the other X active that gives another fur color. So when the animal grows, you're going to have patches of different color because it really depends on which of the two X chromosome was inactivated. So she was right, actually, even though the evidence that she had at the time to show that was actually very small. So it took a lot of work to check this hypothesis, but at least people knew what to look for after that because it matched the data that they had, and they thought, well, if there's a chance for that idea to be right, let's see where that leads. And so one of the most famous examples is the three-color cats, the calico cats. When a cat looks like this, it's always a female. When a cat has got three colors, it's a female because one of the genes that gives the color of the fur is on the X. And so when they look like this is because one of the gene has been inactivated in some patches of cells. And so that's why the cat looks patchy, because the females of mammals are always chimera in the genetic sense. Okay, that means that the cells in the female are not identical genetically because there are two chromosomes, but only one of them is active. So genetically, like they either have one that's active with all the genotypes, all the SNPs, etc. on that chromosome, or the other one, but not both. Okay, males in comparison don't have this process happening. They have only one chromosome that's active, so they're uniform in terms of the cells in the body. So the status of the X, active or inactive, is epigenetic, and that's what's really important in that system. So the way it works is that the zygote here with one cell has got two active chromosomes, so it's usually written M and P for maternal and paternal. So they put the P in pink for some reason, even though that's paternal, and the M in blue, even though that's maternal, but never mind. So in the zygote, that's two active chromosomes, then they make a few divisions, and you still have like an embryo with few cells, let's say 32 or 64 cells that are where both chromosomes are active. At some point, the X inactivation process happens, and you keep either one active or the other active. Okay, so that's the random inactivation process. So far, so good. We'll see how that works in a minute. But what's really important is that once it's chosen, all the descendants of that cell here will keep the same chromosome active or inactive all the time. You decide only once, and after this, you keep a memory of that decision. So that's really going back to the idea of Conrad Waddington. Once you've gone to one path, you've gone that path forever. You cannot just switch branch anymore. Okay, like the whole clone of cells will keep the same chromosome active or inactive. So after inactivation, the same X chromosome, XM or XP, for maternal or paternal, remains inactivated in the descent of the cell. This creates patches of cells where the XM is active and others where the XP is active. Okay, so that's what they want to represent here, is that if that happens early on, let's say 32 or 64 cells, you're going to have a patchwork of different kinds of cells. In the germline, the inactive X is reactivated so that all the oocytes have an active X. Okay, so like in the body of females, there's only one X that's active at all times, but for reproduction purposes, they reactivate both so that in the next generation, everybody receives an active X. Like they would not just keep the X in the inactive status and pass it to the next generation. So this kind of inactivation process is really important to understand diseases. So one example is the Rett syndrome. It affects only girls, and it is due to a mutation on the X-linked gene, which is called MECP2. So MECP2 is an essential gene, so mutation is lethal for boys. So imagine, like they have only one X chromosome, that gene here is affected, so they can't even live. The embryo is barely formed. So usually people don't even notice that they're pregnant when that happens, so you don't even notice it. Boys cannot be born with that mutation. For girls, the mutation is in half of the cells. Okay, so you have one X that's fine, one X that's not fine. So because of the inactivation process, there's a random number of cells that are fine, that don't have the mutation in practice, and that allows them to survive. So they look like this. Like this is an example of a person who's not too much affected, and so they have mental retardation, malformation, and the characteristic hand position. They always hold their hands like that, always. So like there's something in the brain that makes it like deterministic, and that's one of the most characteristic features of the Rett syndrome is really the positions of the hands, which are always like that, as you can see on that picture here. So the inactivation of the X chromosome process is really important to understand genetic diseases, to understand why it's the opposite of what you learn in genetics, that for hemophilia, for instance, or color blindness, is the males that are sick and the females are almost never sick. In some cases, the reverse. When the gene is really important, males cannot have it because they're not even born. But females can have some tolerance with it because only half of their cells have the mutations. So what's the major difficulty then with that system? Like it seems fine. There's a question. Shouldn't the cells with the MSCP2 mutation die early in the development of girls, making them homozygous for wild type X like boys? It's an excellent question. And obviously that's not really what happens. I think a bit, but the issue is that in some cells they don't die. The cells are just dysfunctional. Right? Some cells absolutely require it. In that case, like the cell, if there's a determination that this cell would be the one that has the mutation, it would die and be replaced by a cell that does not have the mutation. But for some other cells, it just makes the cell not functional and not die. And this is what's actually a big problem in that case. You'd rather not have the problem at all, but because there's a tolerance for the problem, then that's what allows it to be in the body. I hope that answers the question. So what's the problem? What's the difficulty with that system here? So think about it. We have said that we have two identical X chromosomes in the cell. They don't have exactly the same DNA sequence, but they could. All right. And we're saying that one of them is inactive and the other one is active. Fine. Why not? Also, we say it could be random. That could be this one or that one. Like we don't know that. So what's the problem now? How to explain that only one chromosome responds to transcription factors, activators or repressors? If you remember what you know on transcription, the way it works is that you've got transcription factors that are proteins that just find the DNA sequences, bind to them, and either activate them or repress them. If you say I've got two X chromosomes here, so any transcription factor should be able to activate one and activate the other, or repress one and repress the other. So either way, there should not be any difference between these two sequences. Okay, if transcription really works by transcription factors, you really can't explain that a whole chromosome would just shut up while the other one would remain active. Okay, whatever activates this one should activate the other one because they got the same sequence or almost the same sequence, especially if it can be random. Okay, so any repressor that binds like a transcription factor binding site that either represses transcription or activates it, if it's an activator, should act on both chromosomes identically. There should not be a difference. So that thing here is impossible to explain with a classical model of transcription. Okay, this does not fit with a classical theory of transcription. So that's something that's really important to understand in full, because it's a major mismatch with the modern theory of transcription through transcription factors. I hope you guys see it, right? That this is not trivial at all, that something would work, that you could just inactivate a sequence here, because anything that inactivates it should inactivate the other chromosome as well. So it has to be something that's epigenetic. It has to be something that keeps the memory that one of the chromosomes has been inactivated, and it's always going to be this one, and you never switch to the other one, or you don't reactivate both. And that mark is DNA methylation, cytosine methylation, something that we've seen before when we spoke about evolution. So cytosine methylation was proposed, again, purely hypothetical, in 1975 by Riggs, to be the mark that differentiates the active X chromosome from the inactive one. So cytosine methylation, remember what that is. That's a cytosine here, and in position 5 of the cycle, so 1, 2, 3, 4, 5, you got this CH3 there, okay, which is usually not present for a cytosine. So you can put a methyl group, 1 carbon, 3 hydrogen, in position 5 here, and that creates methylated cytosine, also known as methyl cytosine. And we've said, if you remember when we mentioned that for evolution, that the CPGs, the CGs in the genome, are usually on the promoters, and they are not methylated, because methylation represses transcription. And so this is why when you look at the human genome, or mouse genome, or any mammalian genome, you see that the CG dinucleotide is much lower than expected. It's 1 in 100 instead of 1 in 16. Except in these so-called CPG islands, which are there, where you have the normal expected 1 over 16. And I said that the reason why the CPG islands can exist is because they are not methylated. Because if they were, that would inhibit transcription. So it's the same system, the one that's active on the X inactivation process. It's through this epigenetic mark, which is methylated cytosine. So when you look at the DNA methylation status of the active X and the inactive X, this is what you see. So you got these genes here, which are represented by these boxes. So on the active X, the one that has transcription, if you look at the promoters, they are not methylated. These lollipops here, which are white, is standard in the field for non-methylated. And the ones which are black, also standard for being methylated. So they represent CPGs that are either methylated or not. And you can see that there is a bigger cluster of CPGs at the promoter. That's what I just said. Promoters have got more CGs than the rest of the genome. So the promoters on the inactive X are not methylated. The rest of the genome is methylated, of the chromosome, I meant. Okay, so inside the genes, you have methylation. On the inactive X, it's the reverse. The promoters of the genes are methylated. There's these SKP genes, the ones that are, there are very few. We're talking of, let's say, 10 to 15 that are active on both the active X and the inactive X. So these ones don't have methylation of the promoters. And the other usual normal genes, they're methylated. Their promoters are methylated, but the bodies of the genes are actually less methylated. So you can typically see results that look like this. There's one gene, PDK3 here, and people are doing a methylation assay on that particular part, and they represent again the open circles for no methylation, and the closed circles, therefore, yes methylation. And so they can distinguish the active chromosome from the inactive chromosome in these kind of experiments. And you could see that there is approximately 5% methylation here on the active chromosome and 76% methylation of the inactive chromosome. So the same sequence will be much more methylated on the inactive X than on the active X. And these sequences are systematically in promoters. So the promoters of the genes in the inactive X are methylated. Those of the active X are not methylated. The assay on the left is called bisulfite sequencing. Bisulfite converts every C to a T unless it is methylated. This allows to know which cytosines are methylated. That's a relatively straightforward assay. Treat the DNA with bisulfite. If the C is methylated, it's going to remain a methylated C. If it was not methylated, it's going to turn into a T. And then by just sequencing the result, you can know whether this cytosine was methylated or not. So typically the results look like this, and you have this very big divide between the inactive X, which is methylated, the promoters are not methylated, and the active X in which the promoters are not methylated. So the most important question is, why does methylation sticks? Like in the clone of cell, like the inactive X is going to remain the same inactive X, not going to switch to the other one. So it means that the inactive X is always going to also pass the methylation status to the daughter cells. How does that work? The enzyme DNMT1 for DNA methyltransferase 1 travels with the replication fork and maintains methylation on the CPGs that are hemimethylated. So you have the replication fork here that opens. You got the leading strand, and so the one with the that's the Okazaki fragments here. That's the leading strand, and so it's going to go this direction here at the bottom. You will have to replicate the CGs that are here on the right. And so as soon as you have replicated the DNA, you have a CG here on the mother strand, which is methylated, but on the daughter strand, CG is a palindrome, so that's also CG if you read it from the other direction. Okay, from 5' to 3', you got 5' CG 3', and on the daughter strand is 5' CG 3'. Okay, so every CG that is methylated on the mother strand will be also methylated on the daughter strand, because there's this enzyme DNMT1 that's traveling together with the replication fork, with the DNA polymerase, actually not with the replication fork, that completes the DNA that is hemimethylated. Okay, so this one here, they just mean that it's been missed, like there was a CG with a methyl group on the parent strand, and then the DNMT1 missed that C here. Usually it does not, but that happens. Okay, you see this example, right? For some time, here this is another example. For some time they were hemimethylated, which means only the parental strand has got the methyl group, but the daughter strand hasn't yet had time to receive the methyl group. And that's the job of the enzyme DNMT1, to complete these methyl groups that are missing. It does not modify the CPGs that are not methylated at all. Okay, when the CG has got no methyl, like this one here, when the replication fork goes through, it's just going to leave it as is. It's not going to put new methyl group. It cannot. It can only complete hemimethylated to fully methylated. So if a CPG is methylated, it remains methylated in all the daughter cells. Okay, because with that process here, there's half of the signal that's on each strand of DNA, but then the DNMT1 is filling the methylation status, so that both daughters now have fully methylated CGs every time. I hope that makes sense for you, that there's this mechanism of copying the information from the parental strand to the daughter strand. So that tells us how the DNA methylation can go from the inactive X to the daughter cell in both inactive X of the daughter cell. But that doesn't tell us why the inactive X is not transcribed. Okay, the promoters are methylated, but why does that stop transcription? Well, the protein MECP2 actually recognizes methylated CPGs and recruits very strong transcription inhibitors. So that's the MBD domain here of MECP2, that's DNA, as you recognize here. What's really hard to see on this picture is that there are two chemical groups here that are very difficult to see. They're in the back, and there are the methyl groups, and they're on the cytosines that are there. And the loop here in green of the MBD domain of MECP2 really recognizes these two methyl groups there. And so that protein MECP2 has the capacity to directly interact and recognize every methylated CPG in the genome. And MECP2 itself, once it's bound, will bring other proteins that repress transcription. Okay, so like you've got the CPG addons here with the methylated CPGs, the MBD of MECP2 is recognizing them, and then that brings plenty of stuff to just repress transcription. If you have no methylation on the CPG allenes, then the complex here with MECP2 is not binding, and there's no repression of transcription. They can be activated. Okay, so it binds MECPGs with a domain called MBD, methyl binding domain. So MECP2 is a member of the MBD family of proteins. It's the first member of the MBD family. MECP1 appeared for a short time in the literature, and then it turned out to be an artifact. So the very first member was MECP2. And TRD stands for transcriptional repressor domain, the one that's here there. MECP2 is indeed the same gene that is mutated in the Rett syndrome. Okay, so that has nothing to do with what I said before. It's just that that gene happens to be very important for the inactivation of the X, and in parallel to that, to be important for the Rett syndrome. So now we understand a little bit better how once you've got methyl groups, it's going to shut down transcription because these methyl groups are sticking out of the DNA. Some proteins can recognize them and initiate a mechanism of shutting off transcription. But how do you choose which chromosome is going to be the lucky one or the unlucky one? So the gene exists for XI specific transcripts, so XI is inactive X, that's the way they call it in the field, is a long non-coding RNA that is expressed only on the inactive X. It coats the whole chromosome and spreads the signal to methylate the promoters of the genes. So initially you've got two X chromosomes that are both active, and one of them starts to transcribe the exist gene. Okay, it starts to activate the transcription of that gene here, and that's the RNA that's here. And that RNA, for a reason that's not fully understood, is going to creep along the whole chromosome and to coat it completely with that RNA here. And that RNA is the signal that says, please methylate the promoters here and here and here and there. Okay, that's just one of the functions, the main function of the exist long non-coding RNA. And after the process is finished, one of the two chromosomes is just like as new, nothing has happened, and the other one has been like completely coated by exist and completely methylated in terms of the promoters. So when you look at it with immunofluorescence, you can use, for example, RNA fish to see RNA, and the inactive X has got a lot of this exist RNA, which is there, highlighted in red. The exist locus is here in yellow, and you can see that there are two loci, of course, because there are two chromosomes, but only one of them is like completely coated in this exist RNA. So on the active X, exist is the only expressed gene, okay, like because it shuts down all the rest. On the active X, every gene is expressed except exist. Okay, that's not exactly true because there are the escapees, right? But that's the idea. Either you express exist that really shuts down the whole chromosome, or you don't express exists and then you can express the whole chromosome. So it's really important to understand that this is epigenetic because there is an epigenetic mechanism, and that epigenetic mechanism is what DNMT1 does. The case of CPG methylation is clearly epigenetic because the enzyme DNMT1 copies the mark on the daughter strands to maintain the information in both daughter cells. Okay, people don't really know what they mean when they say epigenetic, but there's this idea of a memory, and the memory of the event is DNA methylation. That's the one thing that's passed from one cell to the next. Okay, whatever happened 15 generations ago, the signal is still there because DNMT1 has faithfully copied the information on all the daughter strands. So that's what makes it a memory process. That's what makes it epigenetic, and we have the mechanism that makes it epigenetic. And that's why I said that X inactivation is a true canonical epigenetic process. We fully understand why the cells will keep the same inactive X in their progeny, and we also understand why transcription will only happen in one of the two chromosomes. The one that is just like as new transcription factors can go and activate it. The one that has been methylated, transcription factors are overruled by DNA methylation. Okay, like the MECP2 is stronger, it trumps the transcription factors, so it's able to kind of keep them at bay, and they are no longer able to activate the inactivates. Do we know how the cells select the methylation, that methylation will happen at specific genes on that inactivated X chromosome, or not all the genes? Is that yet to be discovered? So there are the SKPs, remember, like these 10-15 genes that are not inactivated by the during the process, and no, I don't think that this is really well understood. So exist doesn't seem to get to them somehow. So they have some kind of, resistance mechanism which has not been worked out just yet. But the default is like inactivate all the genes. People have tried that, so they put exist on another chromosome, like chromosome 21, for instance, and then that will inactivate the whole chromosome 21 without problem. So once exist is there, it just creeps and just shuts down everything. But there exists this kind of protection mechanism for the SKP, which is not understood. Why does exist RNA not affect the active X chromosome? Well, why does it not like travel and then go to the other chromosome? I don't know. It's a very good question. So what makes it stick and stay on only one chromosome? I got no idea. How does it even know it's the same X chromosome? And because the chromosome is quite long, it was transcribed here, and so when it's over there, how does it even know that the transcription site was there? I don't know. Maybe people know, that's not exactly my field, but I think this is not fully understood. Okay, so it's time for a break. It's 3:01. We're going to restart in 3:08. I don't have a lot of things to say after that, so enjoy your break until 3:08. Okay, we're back. Are there any questions so far? Things you'd like me to explain again or to go deeper into? Or was everything clear? It's clear. Nice. So let's go back a little bit now to some general theory to see a little bit how this is interpreted in general by people in the field. So the model is the writer, eraser, reader model, WE or view of epigenetic. Epigenetic pathways are decomposed into writers, erasers, and readers. We have seen the writer DNMT1 and the reader MECP2. Okay, so you have a signal, that's DNA methylation in that case, a CpG methylation. So the idea is, if you want something to be epigenetic, it has to stay somewhere. It has to remain, the information has to be there and pass to the next cell. So that information has to be put, that's for sure. That information has to be able to be erased. If you can only put the information and there's no information at some point, the information is everywhere, and if you cannot discriminate, then you have no information. So if you can put it, you have to be able to erase it somehow, but you also have to be able to read it, and that's kind of an important component, is that this information per se is just a signal. This is not something that has an action by itself. There has to be an interpreter that's able to do something with that information. So the first writer that we've seen is DNMT1. That's the one that takes half of the information and then writes the other half during the cell, during the division, when the genome is replicated. We haven't seen any eraser in that process, but still there must be a process, because I said that the inactive X gets reactivated in the germline. And one reader that we've seen is MECP2. That's the protein that can detect the signal of methylation, just interacts with it specifically, and then is able to do stuff after that, like repress transcription. Okay, so whenever we think of epigenetics, we think of at least three components, like a writer, an eraser, and a reader. And we can't consider that we understand what's going on until we have these three components at least. We have seen the writer DNMT1 and the reader MSCP2, but there are many more components involved in the epigenetic regulations of CpG methylation. So I'm showing you other players that are known in like 4 CpG methylations. So you have 2 other DNA methyltransferases like DNMT3A and 3B. There are the erasers TET1 and TET2, and the readers, there are really a lot, like MBD2, Kaizo, UHRF1, and there's a very long list of readers that can interpret DNA methylation. So you have relatively few writers, like 3 in that case, and one of them is this very specific writer. DNMT1 is, in a sense, the most important because that's the one that gives the epigenetic nature to the signal. That's the one that maintains the signal. And DNMT3A and 3B, they are the de novo methyltransferases. They are the ones that once when exist comes and starts to coat the chromosome, it brings these de novo methyltransferases. It says I want to convert a non-methylated CpG to a fully methylated CpG, and for this you need one of these 2 there. Okay, so that's the standard view for every epigenetic mechanism, that there has to be a couple of actors that are always the same. But CpG methylation is not the only thing that's considered epigenetic, and definitely not the only part of the epigenome. Another part that you probably have heard about, because it's already quite famous, is the epigenetic modification of histones. So histone tails can be modified by methylation or by other types of chemical modifications. They can be acetylated, phosphorylated, and sumoylated, you name it. There's really a lot of potential modifications. There are many writers, erasers, and readers for each modification. Okay, so it's not just about the DNA being modified. Like DNA, of course, is really very close to the genome because it's really on the DNA of the genome, and the histones, they're very close as well. And there are much more possibilities for writing a signal on their tails here. So nucleosomes are usually represented as these cylinders around which the DNA wraps approximately 2 times. And the histones have got non-structured tails that protrude out of the nucleosome, and it's as if like there was a little piece of paper so that a writer could write something, can erase it as well, and that you'd have specific readers that will be able to interpret whatever is written on that piece of paper. So an important modification that we're going to spend some time on today is lysine methylation. So lysine is part of a protein. It's got nothing to do with DNA. Like cytosine is a nucleic acid, lysine is an amino acid. And lysine methylation is actually very important in the epigenome for this idea of transmitting information from a parent cell to the daughter cell, and for maintaining a sort of cellular memory at all times. So these are the 4 histones here, H2A, H2B, H3, and H4. There's a 5th one, histone H1, which is not part of the nucleosome. Histone H1 is actually separated, and that's mostly used for mitosis, because when it's phosphorylated, it compresses the chromosome much more to make it look like that here. So if you look at the picture of the chromosome that is like visible in microscope, it looks like this. It's made of this solenoid fiber, if you want, even though this is disputed whether that actually exists. You can see it in vitro, but in vivo it's never been really seen. And so at some point you go to this beads on a string model with the nucleosomes here around which the DNA wraps. And if you zoom in even further, you will see the tails with different colors here of the histones on the nucleosome. So here on this map are highlighted the places that can be modified either by acetylation, and that's not what we're going to talk about today, or by methylation, which can be permissive or repressive. That's one way you could put it. And so if you look at histone H3, it has the most possibilities of methylation. Okay, like there's this lysine in position 4, position 9, in position 27, and position 36 that can all be methylated. The other lysines can be acetylated. And for histone H4, this is only the lysine in position 20 that can be methylated. So what does it mean, lysine methylation? It looks like that. So that's a lysine residue here. There is like the radical there with the like the side chain I meant with this NH3 part at the end. So the side chain is relatively long compared to other side chain. And here you can have one methyl group, two methyl group, or three methyl groups. Okay, you exchange these hydrogens that are here for a carbon plus 3 hydrogens. So when it's triple methylated, it looks like that. That's a trimethylated lysine. That's the way it looks at the end. So if you see it, that goes from something that was charged and positive here, so a polar part of the residue, to something that's strongly apolar, like CH3 is like very apolar. So that really changes the nature of the amino acid when it goes from the native state here to the modified state by methylation. And so histone H3, as I said here, can be methylated on 4 key positions, and we're going to see a little bit what these positions are and what they do. So one of these positions is H3K9Me3, and that's important for the formation of heterochromatin. So here, that's a little sketch. You got the 3 methyl groups that are here. That's histone H3, and so H3K9Me3 is jargon in the field for histone H3. That's this part here. K9, so that's the lysine in position 9, and Me3 means that you have put on this lysine 3 methyl groups. Okay, so like the code that people use in the field is H3K9Me3. It speaks for itself. And one thing that you can do to see where that modification is present, for instance, is an immunofluorescence. You have antibodies against that particular modification here, and if you couple them to something that shines in green, when you stain the nucleus of a cell here, and that's a mouse cells, it looks typically like that. You know, there are these bright dots that appear like this, like polka dots, that are very very conspicuous, very noticeable. And if you use DAPI, which is a stain for DNA, so you will see actually high concentration of DNA. Somehow DAPI in mouse cells also highlights more or less the same places like that. That correspond to the centromeres of several chromosomes coming together. Okay, like you have an assemblage of centromeres from different chromosomes that's forming a blob, and that blob here is heterochromatin. So the reason why it's brighter in DAPI is because the concentration of DNA is stronger, like the DNA is compressed, and also more simply because the sequence is AT rich, and DAPI has a bias for A's and T's. But basically, there's this very simple trick in mouse cells to see where the centromeres are. You just stain the nucleus with DAPI, and you can see them directly. When you use these antibodies against that particular modification, so imagine that you have an antibody that recognizes only this trimethylated form here of the lysine, then you see pretty much the same pattern. There's a lot of that histone modification there on the centromeres of the mouse chromosome, and also on the centromeres of other species. You can also see, if you look carefully, that on the outside of the nucleus there is a bit more staining than on the inside. That's also reproducible. Like you will see more H3K9 trimethylation on the outside of the nucleus. So H3K9Me3 is a mark that is abundant in heterochromatin, that is to say the centromeres and the periphery of the nucleus in mammals, where there is no transcription and the chromatin is compact. So it's kind of hard to define what heterochromatin is, but that's a place of the genome where the DNA is really more compact than in euchromatin, the opposite, and where there is little transcription. So centromeres, for example, they're very compressed, very compact, and if you put a gene there, it's not expressed. Like there's really something that kills transcription over there, and one of these things is that particular histone mark here. It's very inhibitory to transcription. If you have this histone marks a lot around a gene, the gene is shut down. So that here is a characteristic of heterochromatin, even though that's kind of arbitrary what you call heterochromatin. This is not arbitrary, and now people define heterochromatin as these places of the genome that has a lot of that particular mark, because that corresponds to what historically they had decided was heterochromatin. Another modification of interest is H3K27Me3. So now we know the code. It means histone H3, lysine at position 27, and 3 methyl groups here. So the 3 methyl groups will be on the lysine at position 27. So H3K27ME3 is a mark that is present on development-related genes and cell identity genes, right? So it's not like H3K9 trimethylation that will go into very large domains like centromeres or like, very big heterochromatin domain. This one is more like surgical, like tiny domains, and that usually coats a gene but doesn't really crawl much further than the gene. And it's even not the entire gene. Very often it's just the promoter of the gene. So this mark is much more, sprinkled over the genome than H3K9 trimethylation, which is smeared. So it maintains an active repression on the genes that must be turned off to maintain a cell identity. So here, when you have H3K27 trimethylation of the histones around the promoter of the gene is maintained off very, very strongly, at least as much as with H3K9, and probably more. That's a more potent inhibitor of transcription. And so that particular state here maintains the gene off, and that's really important to make sure that the gene doesn't suddenly wake up. Remember this example when I was saying in the beginning, a red blood cell lineage will never make neurons. It's also, among others, because the neuron lineage has been completely shut down by this kind of trick here, using H3K27 trimethylation to really inhibit everything that would have to do with anything else than the red blood cell lineage. So as long as the cells keep dividing, then that signal is maintained, and so you have the inheritance of the state off like that, which means that the cells that have started in one lineage, they will just continue in that lineage by default, because the default is just to keep whatever H3K27ME3 was present in one cell to the next. But if you differentiate, it means that you want to change identity, like the cell is going to have a different fate. And in that case, the mark is removed here, and the transcription can go on. So that's a change of identity. Somehow this system here is in reality just a system for inhibiting transcription, but it's been really overloaded very strongly on giving the identity to the cell. So every gene that really contributes to making the identity of a cell is often controlled by that system here, and especially in the early development. So it also contributes to the inactivation of the X chromosome. It reinforces the methylation of CPGs. So the X chromosome is maintained by two mechanisms. So the one that I've showed before, is the methylation of CPGs, but also if you look at a female cell and if you look at this mark, you would see one big blob, contrary to what I've said, which is not surgical but really smeared on the whole chromosome, and that would be the inactive X. It's also coated by that particular modification to make sure that this is very strongly inhibited and there will be no transcription whatsoever except on the escapees. So that here is a generic repression system, but has been really targeted to maintain the identity of the cell. So not every gene is targeted by this, only the cells that are important for identity. Another mark that's very important is H3K4ME3. So histone H3, lysine in position 4, not 27, 3 methyl groups. H3K4ME3 is particularly enriched at the promoters of active genes. It has a complex role and mostly protects against H3K27ME3, except in embryonic stem cells. So when you look at where this mark is present, it really corresponds quite strongly with the transcription start sites, the promoters of the active genes. So it typically looks like that, the amount of signals. So you have, like, instead of having a hill just at the position of the transcription start site, there's a dip and then the end of the hill. So it's a dip on top of the hill. Why is that? It's because at the precise position where transcription starts, there is no nucleosome. That's typically called an NF4, nucleosome-free region, or ND4, nucleosome-depleted region. You'll see this in some of the cartoons. So at the place where transcription starts, there's a gap. There's no nucleosome there. That's the place for the polymerase to come and to start transcribing. So it means that if there's no nucleosome, then there cannot be any nucleosome modification, obviously. And that's why you typically see this signal that should be just a plain hill, but then there's this dip in the middle that reminds us that just there at that location there is no nucleosome, so there cannot be that particular modification. For many years it's been considered to be an active modification, like people said, and you will see this in the literature when you read the papers, especially in the old papers, but even today you will see people say that this mark here is an activator of transcription. When it's there, it helps physically the polymerase come and then transcribe. And this is based on evidence. They didn't come up with this just by chance. It's because there's really an interaction between that particular modification and the RNA polymerase. So people thought the model is quite simple, right? You have that modification which is put on the promoter that really helps, grab the RNA polymerase and load it onto the promoter so that it can start. As simple as that. Turns out it's not as simple as that. The recent evidence that people have tried to acquire is not very consistent with this idea, in the sense that you can have a perfectly fine, perfectly normal cell with a perfectly normal transcription without this mark. Okay, in some cases you can just remove the mark completely and the cell is going to be fine with it. In particular, like ES cells, they're fine. You don't need to have that modification. Their biology is not perturbed. And if you have a cell that is differentiated, not an ES cell, you can remove that mark if you also remove H3K27 trimethylation. Okay, which tells you that the main purpose of this mark is just to prevent H3K27 trimethylation from inhibiting the gene. So it's not really an activating mark, it's more protecting against an inhibition, or as the author put it, protecting against a default inhibition. But you could say that's pretty much the same, and it's not that important whether you call it one way or the other. Okay, so that mark here, a bit more complicated to understand. At the moment there is a bit of a debate about how this could work exactly, and some people say maybe it works more in DNA repair than in transcription, but still you will see this very strong statistical association of the mark here with the beginning of the transcription on the promoters. So extremely strong association. Another mark that's interesting, and that will also bring us to other types of mechanisms, is H3K36 trimethylation. So here again, I unfortunately copy-pasted the same slide as before, so it should be H3, histone H3, lysine in position 36, 3 methyl groups, H3, K36, Me3. So this one has to do a bit with transcription, but it more has to do with other mechanisms, in particular with splicing. And so it's not completely clear how this works, but the idea is the following. You have here a transcribing polymerase that's going from left to right, and of course the DNA is wrapped around the nucleosome, so the transcription has to go through the nucleosomes. The nucleosomes have to be shuffling around, but they stay in the vicinity of the transcribing polymerase. They're not really kicked out completely to go in a different place. They're just passed around the transcribing RNA polymerase. So you have this site here which is indicated as weak PTB site. PTB is a protein, and that's a protein that's important for splicing. Okay, so that's a polypyrimidine like pyrimidine track binding protein. So the site here is quite weak, which means that it will normally not lead to splicing. Like the splicing will be just skipped in that case. And so you have two possibilities in that case. The first is that this mark is present, H3K36 trimethylation is present in the neighborhood, and that will recruit locally these machinery in red here, MRG15 and PTB, doesn't matter what they are. What matters is that they have so MRG15 has a specific interaction with H3K36 trimethylation here, and so once it's there, it's in the neighborhood and it's ready to catch the RNA as it grows out of the polymerase there, to grab it and then make splicing happen. Okay, that is really the critical factor. If you have a strong PTB site, splicing will happen, no problem. But if the site is weak, then it will rely on other signals like this H3K36 trimethylation to actually complete the splicing in that case. If you have no such modification, another one like H3K4 trimethylation, this does not recruit MRG15 and there is no splicing. So the particular exon introns remains and you have a case of intron retention in that case. Okay, like the intron is either skipped or included. And that will depend on the status of this modification in the neighborhood. So when you look at the genome, where do you see that modification? You see it mostly on exons. Every time you have an exon, somehow there's a mechanism, it's not fully understood how that can work, that really marks exons with that particular modification, which means that the cell has a way to mark exons in the DNA, which is very surprising, but yeah, that's pretty much the way it works. At least there's an enrichment for the mark HPK36 on the exons of the genes. Introns have it much less. So that's one of the functions of H3K36. Another one is that it contributes to DNA repair. If you have a double strand break in the vicinity of H3K36 trimethylation, it's going to be repaired faster. That kind of allows the DNA repair to repair the double strand break a little bit faster. And the mechanism is, again, this type of cartoon there where you have the recruitment of specific proteins that interact with HPK36 trimethylation and then come faster on the spot of damage so that they can repair and move on. So these are the 4 examples of modifications of histones that I wanted to talk about today. There are more. There are way more. I've talked only about histone H3. I've not even mentioned the other histones. But I think this is enough to see a little bit the panel of what's out there and what could these potential modifications do. So the idea is that they could play in transcription, they could also play in DNA repair, they could play in splicing, and in pretty much anything that we've looked at, like of course compressing the chromatin. So one question is, how do we know? How do we know that one particular histone modification is present at one location? So there's one standard assay that really is used by almost everybody in the field and that works quite well. And I wanted to show that to you, also because maybe you'll see this when you attend talks or when you read papers. That's a very standard method that became a standard around 2000, a little bit after 2000, more 2005 actually. So the standard method to assay histone modifications on the genome is CHIP or chromatin IP, and IP stands for immunoprecipitation. So that's the standard name. You see ChIP-seq. The main idea is to fix cells and use an antibody against the modification to purify all the DNA sequences that are associated with it. So you have this scheme here, and you're really interested in that histone modification, but that's in green here. And so you can imagine that you fix the cells, like you kill them, and then you use a chemical that really maintains the cell in the shape that they are. So the DNA that's next to the nucleosome is going to remain the way it is. The nucleosomes are not going to go away. You don't need to fix the cells in that case because the interaction between nucleosomes and DNA is just crazy strong. It's one of the strongest biological interactions ever reported. Like the affinity constant in terms of chemistry is like really high. So histones have got a crazy affinity for DNA, so even if you don't fix, actually it's not that much of a problem. The histones are not going to just slide away and disappear. But most people fix. And so once you fix that, use the antibody, which is represented here in purple, and that specifically recognizes the modified lysine of interest. Okay, so this one here, that nucleosome, has not been recognized because it doesn't have the modification. After fixation, here the part that's missing is that you sonicate the DNA. So you use ultrasound to really break the DNA so that you have approximately one nucleosome only, and then you broke the DNA every time you have a separation between the nucleosomes. So that's one nucleosome, another one. The one that's not represented would be this one because it hasn't been recognized by the antibody. And so if you couple this antibody with something heavy, like a very big protein, then you can just go get it with centrifugation because that's heavier than the rest, so they will go to the bottom of the tube. You can also couple it with something magnetic, and then you can use the magnet to really attract the antibody together. You're pulling very much on the nucleosome because it has the modification, but also the DNA. Okay, and the key is that you want to identify the DNA that was in the vicinity of that particular modification. Okay, so I hope it speaks to everybody. Like, you fix the cells you sonicate to separate these nucleosomes with their modification in their DNA. Then use an antibody to kind of, as a fishing line if you want, to go get the nucleosomes that have this modification, and they come with the DNA that's around the nucleosome. So after this, remove proteins, kill them all, and then get the DNA. That's super easy. We have kits for that. So you make a DNA purification, and you don't know exactly what the sequences are here. You got no idea what they are, but you can make an Illumina library. You're experts now with how this works. So you just need to put the P5 and P7 sequences on the side, and you'll be able to just sequence the whole of it using an Illumina sequencer. Okay, so then you got your paired ends, like forward and reverse. They will identify that, that sequence here, that sequence there, but they will leave out this one that is here because you did not precipitate it. Okay, you're going to sequence that, but you're going to remove that. So the sequences that you're going to get at the end are the ones that were very likely to be associated with the modification you're interested in. So that's what I said. You can then prepare an Illumina library and sequence the DNA to get a genome-wide profile of the mark. So the profiles look like this. They're called ChIP-seq for chromatin immunoprecipitation and sequencing. So ChIP-seq profiles, they would typically look like that. So you have the input chromatin here, just to make sure that your antibody is not recognizing some specific sequence or doesn't have an affinity for something else. So you want to see that this is near empty here, and then you do the experiment with an antibody against H3K27AC. So we haven't seen this one, but you can guess, right? This is histone H3, lysine in position 27, acetylation, or H3K4ME2, H3K4ME3, H3K4ME1. So these are the different variants with 1, 2, 3 methyl groups. Now you know the code, so you can pretty much read any of these kind of strings there. And these are transcription factors. So you can see typically the profiles looking like that. So people would represent it this way with a gene here that's SID14 for some reason. It could be anything. You have the scale 20 kb, and if you look at, for example, H3K4 trimethylation, which is the one in red here, you can see that indeed on the promoter there's this very strong peak here of that particular mark. Okay, like the promoter is here, the rest of the gene doesn't have much, but here the promoter is very strongly identified by that particular mark here. All right, so that tells you that statistically in many cells the DNA that's here was associated quite often with that modification here of histone H3, H3K4 trimethylation. If we look at this one, H3K4 monomethylation, well, not that much. Okay, it's not zero, like it can be lower at the 3' end of the gene, but here around there, it's like not super crazy high. And if you look at GATA1, for example, that's really flat. Okay, so that's the way these methods work. I hope that this is clear, that with this antibody trick, you can actually get the DNA, and because the sequencing is quantitative, then the more signal you get here, the more DNA you sequence here, it means the more cells had that particular modification on that particular sequence that is here in the genome. Okay? Stack profiles allow us to get information about the histone modifications and transcription factors that are present on all the loci. So here there's just one locus, but we could do that for more. The epigenome designates the set of histone modifications and DNA methylation on the whole genome. So when you think of all these possible tracks here, on all the possible loci, that is the epigenome. There's not really any simple definition of what the epigenome is, but that's the idea, is whatever you can manage to identify by this method of ChIP, because it tells you that's the way the chromatin has been modified locally, and it doesn't have to be the same in every cell. In some cell, the chromatin would be with such or such modification. In another cell, it'd be another set of modifications. So because it's just above, epi means above, it's just above the genome, like the layer that's really touching the genome. People said that's a good name for that. It's just like the layer just above the genome, the epigenome. So with that, you can do automatic annotations, for instance. So if you keep accumulating many tracks, well, you can do interesting things. With many ChIP-seq profiles, one can assign states to each locus of the genome to discover active promoters, active enhancers, or more. So what has been done around 2010 when these methods became automatic is to say, okay, we're just doing it systematically now. We don't really, care very much about what we're doing. We just want to do it all. So we take H3K4ME3, H3K27AC, H3K4ME1, H3K36ME3, H3K9ME3, H3K27ME3, and you can add more. It's not a problem at all. So you take all the histone modifications you can think of, as long as you got a good antibody for it, and then you do this ChIP-seq experiment. And so that gives you a profile here for each and every antibody that you've got. And the profile just shows, for instance, like for this mark, H3K4ME3, while it's really present on that particular side of the genome, whatever this side of the genome is. There's no scale, but the scale is above here. Okay, so it doesn't really matter what is there, but we know that it has a lot of this particular modification. So you do this for as many histone marks as you can, or as many as you can afford. And once you've got that, you train an algorithm to detect recurrent combinations of these marks. So that's what they represent here. So they have 18 potential states, and they say, for example, we look at these histone modifications, H3K4ME3 till H3K27ME3, and state 1, for instance, would be strong in this one and this one. Okay, it has both H3K4ME3 and H3K27 acetylation. So that defines what they call state 1. We don't know what state 1 is. We just know that quite often we find sites that have just these two modifications, H3K4 trimethylation and H3K27 acetylation. Okay, so you can do that systematically for a certain number of states, in that case 18, and you can see that all of them are sometimes a bit similar but mostly different. Case 6, for instance, has got nothing at all. Like, that's the state that says I couldn't find any modification of any histone in what you gave me. Okay, so they can then identify automatically a certain number of states that are recurrent, and you can see them here. They give them color. Okay, like these states are like active TSS, flanking TSS upstream. At some point when these states have emerged, when the algorithm is finished, you can give them human readable names. Like state number 6 is called weak transcription here. State number 18, which is even more empty than 6, is called quiescent. Look, nothing at all. And once you have this automatic annotation system, you can see that every side of the genome will end up in one color. It's one of the states that is here. So for example, the one here that had H3K4ME3, that's there, it ended up in the state active TSS. And you remember, that's what I told you, like this particular histone modification really is present on the promoters, or TSS stands for transcription start site. It's the same thing. So there's this correspondence between whatever you can see in the epigenome and whatever are the functional mechanism happening at that site. Here in green, this is strong transcription, weak transcription, etc. So that tells you this is the body of the gene, because we find all the marks that are associated with exons, etc. So think about H3K36 trimethylation. That's the one I tell you is associated with exons, and you can see that, yeah, it's not really sharply on exons because the exons are tiny, they're here, but where there are exons, you could see kind of, a hump of that mark there. So that tells you around here is something where there is ongoing transcription. So that's why it ends up in the green states that are here. Okay, so you can imagine that you could do that for not just the transcription start site and the bodies of the gene. You can identify new things like enhancers, other functional elements, potentially sites of recurrent DNA repair. Like whatever happens on the chromosome, whatever happens in the genome, will actually leave a trace in the epigenome, that would locally modify the histone modifications. And if you can read these histone modifications, then you can use machine learning to tell you, okay, at that site, this is what happens. At this other site, this is something else happening. And so you can make automatic annotations of the genome. So automatic genome annotation are done with machine learning methods. So these things, like now, they're too much data for humans to be doing this manually. You need a method that goes automatically, just grabs the data from these ChIP-seq profiles and tells you at that particular place, that's what happens. That's an enhancer because I can find this, this, and that, but I cannot find that. So one question is, are histone marks epigenetic? I've argued in the beginning that DNA methylation is epigenetic because there is a mechanism, and that's DNMT1, that really makes sure that when the cells divide, that there is a replication of the information on both sides there. For a mark to be epigenetic, there has to be a copy mechanism. That's almost the definition of epigenetic upon cell division. In the case of histone marks, the mechanisms have not been fully worked out, and some authors are very critical with this view. So you will see sometimes, especially around 2000 to 2010, there were these opinion articles that said that histone marks cannot be called epigenetic until you have identified the mechanism that will make sure that the mark is faithfully inherited on both daughter cells. So the one that is the most epigenetic is H3K27 trimethylation, the one with cell identity. So in that particular case, I think there are many elements of what the mechanisms can be, but this is not fully worked out, not as much as in the case of DNA methylation. So the term epigenome has replaced epigenetic marks for this reason. So if you read papers from around 2000 to 2015, you will often see epigenetic marks for these modifications of histones. But from 2015, people have started to use more the term epigenome because, first, they know that some people are quite critical with that, and second, they realize that there is a point here. Can you really call them epigenetic? Are they the source of memory from the mother cell to the daughter cell? Or are they themselves the consequence of the true signal for that memory? It's not clear. And so just claiming that this is epigenetic when you don't have yet the mechanism would actually be leading to maybe not making the discovery that you should make, that this is not the true signal that is epigenetic. But still, in people's mind, it seems to be a given that these histone modifications here, once they have been written in place, then there is some memory mechanism that remembers that this modification was here, and in the next generation, when the cells divide, that this signal will be replaced. But that's just a fair warning. We don't really know how that works, and until we know how that works, it's fair enough to not call this epigenetic mark, but just to call them epigenomic mark or epigenome, in the sense that this is not a term that's loaded with this idea of having a memory. So that's it. That's what I wanted to tell you today about the epigenome. So the key points to remember is that the epigenome is a form of cell memory. Remember that very first problem that we had is that the cells have the same DNA and yet they do different things. There has to be something. There has to be a signal that reminds them who they are and what they can be, what they cannot be. And people believe that is somewhere in the epigenome or close to the epigenome. In the case of DNA methylation, that's clear. It's one of the signals that bears that memory. In the case of the histone modifications, it is linked to it, but it's not clear that it is the signal that has the memory in it. X inactivation is a prototype of epigenetic event. In that case, the mechanism has been almost fully worked out from the beginning till the end, and that's a case where we can truly say that this is epigenetic. And histone marks reflect the local function of the genome. So nowadays, people don't care very much whether it's epigenetic or not. They say, it doesn't matter. What happens is that because I have all this readout of all these histone modifications, that also tells me what's happening at the site where I'm making the assay, and then I can guess what kind of biological functions, genetic, genomic functions, are happening just by looking at the histone modifications. And I don't need to know, I don't need to care whether they're epigenetic or not. So that's all I wanted to say about epigenomics for today. So it's time to say goodbye. As a matter of saying goodbye, I would like to tell you that the only knowledge we keep is the one we share, of course, because there's no other way to keep knowledge as a group of people. Of course, you can have it in your head or you can have it in the library, but that's not knowledge that's useful in any way. The only way that we can keep what we know is to make sure that the next generation will also have it. You are the ambassadors of scientific knowledge. It is your privilege to carry forward what you have learned. Be proud of what you have achieved and be generous with it. Never stop learning and never stop sharing what you learn. So thank you very much for being here today, and I'll take, of course, questions if you have any for what we've seen today. So, question from Yiving: is X inactivation the reason why STRs on X chromosome don't have much value in forensics, etc.? I don't think so, in the sense that whether the X is active or inactive is totally irrelevant for genetic identification. Okay, so one thing is really seeing what is in the cell in terms of DNA and seeing what they can do with it. And in terms of forensic, we really don't care what they can do. Like, the cells can even be dead. It doesn't matter very much. You're just using the DNA as a fingerprint, in a sense. Okay, so the reason why there is no STR on the X chromosome, I believe, is because of the difficulty of dealing with males and females. Is that if the sample is male or female, then the number of X chromosomes will be different. And so that's going to just be a little bit too messy. You will see 2 peaks in the case of females if the female is heterozygote, but if the female is homozygote, you see 1 peak, and if the males, you will see 1 peak all the time. So, like, I think it's just because it's a little bit confusing, and there's no reason to. Like, you can just use the rest of the genome for getting the information you want. The Y chromosome is much more interesting for forensics. Goodbye and thank you for the semester. Pleasure. It was a pleasure having you. So you guys still have a bit of work to do. You've seen that I've given 2 extra days for the figures. I apologize to people who had worked hard to just hit the deadline and got really tired and then realized after that, oh, well, I got 2 days, thank you. I could have worked a little bit less. I apologize to these people. And on the other hand, you've done the work. So now you could say you got 2 days, but you can work on something else. So if you're happy with your figures, just keep them as is and use these 2 days to work on something else. Glad to see that it was a good semester. Are you running any graduate labs in the fall? No. I won't be here. I'll probably go to Europe very soon. Thank you guys, you're so generous. Where am I going? To Europe. My wife lives in Europe, actually, so when I teach here, I cannot be with my wife. She doesn't really have a visa here yet. It's complicated. So if I want to see her, I have to go to Europe. She's also an academic. She's finishing her PhD. Yeah, best of luck to her. She needs luck. PhD is difficult at the end for most people. You guys will see this if you do a PhD one day. All right, it doesn't seem like there are no, there are questions. So is the feedback for the blog post anonymous? No, it's not. Do we know whose blog post it is? Yes. Will we know who left the feedback? Yeah, it's not anonymous. Like you will actually, everything is known. It's open, but there's no reason to make it anonymous. Anonymity on the forums is clearly linked to aggression. That's very clear. So you got the anonymous forums online and the ones where you have this reputation system. You can compare, for instance, Reddit, which is anonymous, to Stack Overflow, and you will see the quality of the discussions and the aggressions. On Stack Overflow, it's always extremely polite. People just come here to explain stuff to each other. It's because they're non-anonymous. On Reddit, totally anonymous, you will see that people are pretty bitchy with each other. So I don't believe that making comments anonymous increases the quality, and I do believe that it increases the level of aggression. even though I don't believe that you guys are aggressive in any way, but I just want to keep it very friendly. That's why. Anyway, there's nothing that you should be ashamed of, you know. If there's something that you'd say only if you would say it anonymously, are you sure it's something nice? Right? You have to be proud of what you say, so I don't see that anonymity is really doing a bonus in a sense. Actually, in academia, the way we work is that you probably know that we have to publish papers. By now, everybody of you guys know because you're reading papers, so that's what we have to do. If we don't, we're fired. And these papers are reviewed. So before they're published, there's a period when these are sent to people, to experts in the field, and then they come back with their comments saying, you have to do this, you have to do this, or this study is not very good because of that, or this story is really excellent and you should publish right away. And so this system is anonymous. That's one case where we don't know who the reviewers are. But I refuse to play the game, so I'm always writing my name on the reviews for the reasons that I say. I think that if I have something to say, I'm just saying the argument. You know, whether it's me saying it or someone else saying it, it doesn't make a difference. Either what I'm saying makes sense, or it doesn't. And either way, like I don't need to hide my identity, so that people can contact me if they're not happy with the way that I reviewed them, and that has never happened in approximately 10-15 years. There was never someone who complained directly to me saying I really didn't like your review, etc. So when you remove anonymity, when you make people know who they are, I think that increases the level of discussion overall. That's my opinion. Just an opinion, of course. All right, you guys be good. Try to stay healthy. Don't be sick for the exams. That'd be a shame. So good luck to all, and you'll hear from me very soon for the grades of the figures of the whodunit, and then for the feedback. All right, take care, everybody, and see you.